Hello dear learners, my name is Anubhuti Pandey and today we will learn about plant hormones. So let's begin with our first plant hormone, that is auxin. Discovery of auxin was done by two scientists. First scientists were Charles Darwin and his son Francis Darwin. They observed the auxin in the coleoptile of canary grass. So what exactly they observed was the coleoptile of canary grass bent towards the direction of light. But when they removed the tip of the coleoptile and covered with cap, they observed that there is no more further bending of the coleoptile. So they concluded that there is something that is transmitted from the tip. And later on, auxin was isolated by F.W. Wendt from the tips of coleoptile of oat sibling. Auxin is synthesized by tryptophan. The precursor of synthesis of auxin is tryptophan, and tryptophan work in the presence of zinc ion. There are two types of auxin present around us. First one is natural, which is naturally synthesized in the plant. It is of two types, IAA and IBA, indole acetic acid and indole butyric acid. Synthetic auxins are synthesized in the lab. They are of two types, NAA24T, 245T. So how we exactly observe auxin? What are the experiments that we conduct to learn about auxin? There are two bioassays. First one is Avena curvature test and second one is root growth inhibition test. When we talk about the transport of auxin, the transport of auxin is polar, which means that it is transmitted from the shoot to the roots. Now let's learn about the function of auxin. First function is apical dominance. As we all know that auxin is synthesized in the tip of the plant. So if we want to grow the lateral buds, we have to cut or remove the tip of the plant because auxin inhibits the growth of lateral buds and it promotes the growth of apical buds. Next function is cell division and cell enlargement. So what auxin do is it elongates the cell, it helps the cell to divide, and it also controls the xylem differentiation. Auxin also promote development of roots. It is promoted by IBA and NAA. Next one is prevention of abscission. So when new plant parts are grown, such as new leaves, new buds, and new flowers are synthesized in a plant. So it prevents the falling off of these new plant organs, but it promotes the abscission of older mature leaves and fruits which are no more required in the plant, which are old and which are dried. So it promotes their falling off, but it prevents premature abscission of plant organs. Newly synthesized, newly formed plant organs such as newly formed buds and leaves. Flower initiation, auxin also promote flowering, especially in pineapple and lychee. Parthenocarpy. If we want to produce the seedless fruits and vegetables, we use auxin. We spray IAA. For example, tomato. Seedless tomato is produced by parthenocarpy method, which is promoted by auxin. Next one is selective weed killer. Auxin is also used as herbicide. The most popular herbicide is 2,4-D, which is used to kill dicot weeds, but it has no effect on monocot plants. Potato dormancy. If you want to grow a potato plant, but you don't want to grow it today, you want to grow it afterwards after some time. So you just spray NAA or auxin in the potato tuber, in the potato, so that you can keep the potato tubers in a dormant state. And later you can use that tuber for plant germination, for potato plant germination.